Hello, everyone. Welcome to the Science Public Online Talk Series brought to you by the Faculty of Science at the University of Manitoba. My name is Samar Safi Harv, and on behalf of the faculty, I'd like to welcome all of our viewers online today to the talk by Dr. Margaret Docker from the Department of Biological Sciences at the U of M. Before we start, I'd like to make a few acknowledgements. First, we acknowledge that the University of Manitoba campuses are located on original lands of Anishinaabe, Cree, Oji-Cree, Dakota, and Dene peoples, and on the homeland of the Métis Nation. Would also like to thank the SPOT team shown here, as well as all of our speakers for making the SPOT series accessible to all and possible throughout the summer. And it's my pleasure now to introduce our speaker for today, joining us from U of M, Dr. Margaret Docker. Margaret did her PhD in Fisheries and Aquatic Sciences at the University of Guelph, particularly studying environmental sex determination in lampreys with Professor Bill Beamish. Then she did an extensive postdoctoral training after that, before finally finding her home at the University of Manitoba. Her postdoctoral positions included working as a visiting fellow at Fisheries and Oceans Canada in British Columbia, a visiting scientist at the University of New Hampshire, an instructor and research associate at the University of Northern British Columbia, and a postdoctoral fellow at the University of Windsor. She joined us at the University of Manitoba Department of Biological Sciences in 2006. Docker's current research involves developing and applying environmental DNA techniques for monitoring aquatic invasive species and species at risk. This would allow the detection of a species' presence based on the DNA it leaves behind in the water. Margaret's talk will be for 20 minutes, followed by a question-answer period for another 20 minutes. To enter your questions, please go to slido.com and enter the event code 2521 shown on your screen, and we'll be playing this throughout the talk. Feel free to enter your question during the talk itself. I'll be moderating the questions, and you'll be able to see them also after the talk is over. So with that, Margaret, over to you. Welcome.
Okay, well, it's a pleasure to be able to talk to you this afternoon about lampreys, a generally underappreciated group of ancient jawless vertebrates. I'm going to try to keep the talk light this afternoon, but I'm hoping that you'll come away from it with a new appreciation for lampreys, the good and the bad. Lampreys, along with hagfishes, are one of two surviving groups of jawless vertebrates. And so by jawless vertebrates, they have mouths, they have rasping teeth, but they don't have the hinged jaws that we see in all the other surviving vertebrates. Lamprey fossils, the oldest known lamprey fossils, date to about 360 million years ago, but the group is likely at least 400 or 500 million years old. This means that lampreys have survived through at least 4 mass extinction events, including the most recent and maybe the best known of the mass extinction events that wiped out the dinosaurs 65 million years ago. So this means that hagfishes and lampreys represent the dawn of vertebrate evolution. There were other jawless fishes that existed, many other groups, but they're all extinct now. And so hagfishes and lampreys are the sole survivors from this early stage in vertebrate evolution. And so they're often said to sort of represent living fossils from a period in vertebrate evolution that is poorly represented in the fossil record. So studying hagfishes and lampreys is shedding a lot of light on this origin of vertebrate complexity, what's often said to represent one of the major jumps in animal evolution. So there are about 45 surviving lamprey species worldwide, found in the cooler regions of both the northern and the southern hemispheres. Some of them include the Pacific lamprey, shown here, found on both sides of the Pacific Ocean. One of my favorites, the pouched lamprey from the southern hemisphere, which has this beautiful iridescent blue color as a juvenile. And another of my favorites from my PhD work is the tiny least brook lamprey. This is an adult pictured here, and that's as big as it gets. So lampreys have a very interesting and a complex life cycle. The larval stage is very prolonged, lasting several years. The larvae burrow in the sediment of rivers and streams. They're blind at this stage. They're toothless. They look almost more worm-like than like a vertebrate. They burrow in the sediment with their heads sort of partially sticking out, filter feeding detritus and algae from the water column. Because they look so different than the adult form, for the longest time, they were thought to be a separate animal altogether. It wasn't until the late 1800s when someone discovered that these larval creatures actually went through this very dramatic metamorphosis and became adult lampreys, very similar to a caterpillar turning into a butterfly. So during this very dramatic metamorphosis, virtually all of the larval tissues and organs are remodeled into the adult form. And so you see the vertebrate eye becoming very apparent and functional. The filter feeding mouth now becomes that oral disc with the suction cup-like mouth and the teeth, and they become much more mobile at this stage. 18 of the extant surviving species are parasitic after metamorphosis, so they usually migrate downstream, leaving the river and migrating out to lakes or oceans, where they attach to and feed on the blood or flesh of other fishes. This stage lasts from a few months to a few years. They grow very quickly during this stage, after which they will then return to the rivers, migrate upstream, and spawn and die. All lampreys die after spawning, very much like Pacific salmon do, although unlike Pacific salmon, lampreys don't return to their natal stream. If you're at all familiar with lampreys, chances are that the one that you're familiar with is the invasive sea lamprey in the Laurentian Great Lakes. Certainly in North America, this is the one that gets most of our attention because it has been such a problem in the Great Lakes. As the name suggests, sea lamprey, the sea lamprey is native to the ocean. It's found on both sides of the Atlantic Ocean, where it occurs at very low densities and feeds on large-bodied fish and sometimes marine mammals. So it's not a pest in its native environment. It is sort of lives a peaceful coexistence with its hosts. Where it is a pest, of course, is in the Great Lakes. Like many aquatic invasive species, once the sea lamprey gained access to the large, rich freshwater resource of the Great Lakes, it became a huge problem. There's a little bit of debate about the actual sort of timing and route of invasion, but sea lamprey likely got into first Lake Ontario in the mid to late 1800s through man-made canals coming in from the Atlantic Ocean. After that point, with the building of the Welland Ship Canal that allowed boats to bypass Niagara Falls, this canal also allowed sea lamprey to get past Niagara Falls. And so then sea lamprey was able to colonize first Lake Erie, and then it rapidly made its way into the upper Great Lakes, Huron, Michigan, and finally Superior, so that sea lamprey was well established in all of the Great Lakes by the end of the 1930s. So even though not a pest in its native environment, sea lamprey became a huge problem in the Great Lakes, the main contributor to the crash of the lake trout populations, and was largely responsible for decimation of the multi-billion-dollar commercial and recreational fisheries. So a binational joint Canadian-American Great Lakes Fishery Commission was formed in 1955 and established a sea lamprey control program to try to control the sea lamprey and rehabilitate the Great Lakes ecosystem. The Sea Lamprey Control Program is one of the largest pest control programs ever mounted against a vertebrate pest, and it relies largely on sort of two methods to control the sea lamprey: barrier dams, which prevent sea lamprey adults from accessing the rivers and reproducing, and a pesticide that's applied to the rivers that selectively kills larval lampreys to prevent them from eventually metamorphosing and feeding on the fish. The whole story of the Sea Lamprey Control Program is really interesting. I'm happy to answer some questions at the end of the talk, including about my own research related to Sea Lamprey control. I will also direct you to a recent book by Corey Brandt that details the sort of fascinating story of the invasion and control of sea lamprey in the Great Lakes. What I want to focus on for the rest of my talk, though, is some of the good lampreys, the ones that are probably underrepresented and underappreciated. And so I wanted to talk about some of these good native lampreys, including the 3 species that occur in Manitoba: the silver lamprey and the chestnut lamprey, 2 parasitic species, but again, they co-evolved with their hosts so that they're not considered pests. And then the 3rd species found in Manitoba is the tiny northern brook lamprey. The northern brook lamprey is one of 20 lamprey species that are called non-parasitic. That means that they don't feed at all after metamorphosis. So that in these non-parasitic lampreys, we have the same larval stage that we saw in the parasitic species, where they're blind, toothless filter feeders in the river sediments for several years. They undergo the same metamorphosis that we see in the parasitic species, but this is where the pathways of the 2 different types of lampreys diverge. The non-parasitic species completely bypass the adult feeding phase, going directly to sexual maturation without feeding at all on fishes, or feeding at all, period. And so they mature at very small sizes, in fact smaller than the largest larvae, because they shrink quite a bit during that 6 to 10 month non-trophic period. They also die after spawning. So hopefully you're starting to gain something of an appreciation for lampreys, and in fact throughout most of the world there is a long established appreciation for lampreys. And so, for example, many of the Native American tribes in the Pacific Northwest have long appreciated Pacific lamprey. So the Pacific lamprey, known as the first food, has been used as food and for medicinal and ceremonial purposes for millennia. And the Maori in New Zealand have also long appreciated the pouched lamprey. Same in Europe. The ancient Romans certainly appreciated lampreys. In fact, one of the ways that you could curry favor with Caesar was to present him with a gift of lampreys, and the same applies in medieval Europe. Medieval England, some of you may have heard the tale of Henry I of England, who is said to have died after eating what was described as a surfeit of lampreys. Continuing on to present day, the lamprey pie is an important staple during coronations and other special events in the royal family, and apparently lamprey pies are even mentioned in the HBO series Game of Thrones. Elsewhere in Europe, again, lamprey is highly appreciated. Lampreys appear on the coat of arms of some municipalities in Spain, and we see lampreys on postage stamps in Finland as well. Lampreys also have considerable ecological significance. We're certainly most aware of them when they're predators, particularly when they're on predators that fish predators on fish that we're interested in, but they also have really important functions at the base of the food chain as well. Larval lampreys, really important in nutrient cycling because they feed on detritus that is not really digestible by most other fishes. They're really important in taking that detritus and converting it into larval tissue that can then be fed on by other fishes, for example, on sturgeons. Also really important prey at other points in their life cycle. Downstream migrants, for example, after metamorphosis, when they're leaving the streams, often in large pulses, they're fed upon by a number of other predators. For example, sea lions that will wait at the mouth of rivers for those downstream migrants to come out. And then, of course, after they've fed, become large and fat, and they're returning to spawn in rivers, they're also fed upon by a number of aerial, aquatic, and terrestrial predators. In addition to their role in the food chain, they also have a really important role in what is known often as ecosystem engineers. And so the burrowing activity of larval lampreys and then the nest building activity of the adult lampreys make changes to the stream ecosystem in ways that benefit other stream organisms, such as a number of macroinvertebrates. And then, if that isn't enough, even after death, the carcasses of anadromous lampreys, those that feed at sea, are a really important source of marine-derived nutrients, bringing nutrients from the sea into nutrient-poor fresh water. And then again, I'm just trying to continue sort of heaping praise on lampreys. Lampreys are also becoming really significant in biomedical research. And I'll give you 4 examples. One is lampreys are becoming a really important model for studying spinal cord regeneration in humans. Lampreys are the only vertebrates known where the spinal cord can be completely transected and they're able to regain full function. Lampreys are also being used as models to study biliary atresia in human infants. So in human infants with this disorder, they're born without a common bile duct so that they're unable to eliminate bile into the small intestine. This means that they begin to suffer jaundice, malabsorption of nutrients, cirrhosis of the liver, and this condition is eventually fatal if a liver transplant isn't possible. Lampreys are an important model to study biliary atresia because, as it turns out, all lampreys lose their gallbladder and bile duct as a normal part of metamorphosis, and they're able to live in some species for several years without these structures. So they're using to help inform the condition in human infants. A third area of biomedical research in which lampreys are used is the search for novel anticoagulants to treat blood coagulation disorders and strokes. So blood-sucking insects and other invertebrates, such as medicinal leeches, have sort of long fascinated scientists as a source of anticoagulants. And the salivary gland secretions of parasitic lampreys are now being mined as well as a possible source of novel anticoagulants, local anesthetics coming from the enzymes that parasitic lampreys use to anesthetize the host, and immunosuppressants that lampreys use to suppress the immune response of their host. And then a final biomedical application of lampreys is lampreys are also helping to inform a human condition known as iron loading. That blood-feeding lampreys, blood is a really rich, easily digestible nutrient, but the downside of feeding so heavily on blood is the accumulation of iron. So other blood-feeding animals, such as vampire bats, they also take in a lot of iron in the diet, but these other blood-feeding animals limit the amount of absorption. Parasitic lampreys don't limit the uptake of iron. They take it all up, and they're able to actually store and tolerate very high concentrations of iron, sometimes orders of magnitude what would be toxic to other vertebrates. And so lampreys then are being used to help understand hemochromatosis in humans, where humans continue to absorb iron after their requirements are met. So despite the the huge problems of sea lamprey in the Great Lakes, elsewhere in the world, conservation of native lampreys is becoming increasingly important, certainly in the Pacific Northwest and New Zealand, and in Europe as well, and increasingly in parts of North America as well. A lot of these lampreys are at risk from a variety of anthropogenic pressures, such as dams that act as barriers to migration during that complex life cycle, pollution, habitat destruction, particularly the dredging and removal of those larval sediments, and in some cases as well, overharvest. Quite ironically, conservation concerns exist for sea lamprey as well in its native range. And so the same species wreaks havoc in the Great Lakes, but in Europe, it's highly valued and it's a protected species. And ironically, in 2017, pink salmon were found to have invaded some Scottish rivers, and there's actually concerns there that these invasive pink salmon will negatively impact the native sea lamprey by potentially disturbing its nest. So kind of the reverse of what we're seeing in the Great Lakes. So what about the native lampreys in Manitoba? I mentioned that we have 3 species here: the northern brook lamprey, chestnut lamprey, and silver lamprey. As it currently stands, all 3 species have been assessed by CSIWIC, the Committee on the Status of Endangered Wildlife in Canada, as being data deficient. And this is due to a lack of information regarding their distribution and abundance in the province. And it's not really known whether the relatively few records that we have to date are due to a real sort of patchy or restricted distribution, or it's just because they haven't really been noticed before, particularly because that long larval stage is spent burrowed in the sediment, that unless you're doing targeted sampling for lampreys, it's quite possible that they've largely gone undetected so far. So I'll just very briefly talk a little bit about what we've learned about their distribution in Manitoba. Recently, my lab has used a relatively new technique, environmental DNA, to help sort of fill some of these gaps about the distribution of lampreys in Manitoba. So environmental DNA, or eDNA, is the DNA of an organism that persists in the environment. So from mucus, feces, sloughed off cells, it can be collected in a simple water sample, filtered, and then using genetic techniques to identify the species of origin. Because it is so sensitive, it's really been very helpful in detecting rare aquatic species, either species of conservation concern or invasive species at the leading edge of the invasion. So I'll just very, very quickly then sort of tell you a little bit of what we have learned or been able to confirm with the 3 species in Manitoba. So the chestnut lamprey, it's the most widespread of the 3 species in Manitoba, quite widespread throughout southern Manitoba. The Red River and its tributaries, Assiniboine River and tributaries of Lake Winnipeg. If you want to see a chestnut lamprey, probably one of the best opportunities to see them is when they're spawning. They spawn during daylight hours in relatively shallow streams, usually when the water temperature reaches about 17 degrees Celsius, so that's usually sort of the last 2 weeks of June. They're about 2 feet in length at maturity, so usually quite visible at that point. The other time to spot them in Manitoba, if you're an angler, is when they're attached to fish during the parasitic feeding phase. Chestnut lamprey, like most parasitic lampreys, aren't particularly choosy, and they have been found feeding on a large number of species, and so that would be another good opportunity to spot them. Silver lamprey has a somewhat more restricted distribution in Manitoba, found predominantly, or at least recorded predominantly, in the Winnipeg River watershed, and then further north in the Nelson River watershed. Silver lamprey tend to be a little bit bigger at maturity than chestnut lamprey, reaching about 30 centimeters in length in Manitoba, although they do get quite a bit larger elsewhere in their range. Silver lamprey, again, not particularly choosy. You're going to be able to find them attached to fish in the province. Again, walleye, suckers, and other smaller bodied fish, although silver lamprey are particularly fond of larger bodied fish, in particular lake sturgeon and northern pike. And in fact, the only silver lamprey I've ever seen in the province have been ones that have been collected on fish. Unlike most other lampreys that spawn in shallow water, silver lamprey tends to be a little bit more cryptic at other stages of its life cycle. At least in Manitoba, seems to spawn in deeper waters, not those wadable streams that we saw for the chestnut lamprey. And it's probably the best guess at where they're spawning in the Winnipeg River is at the aptly named Lamprey Rapids. And then the final and third and final species in Manitoba is the northern brook lamprey, one of those non-parasitic species. Because it doesn't feed at all after metamorphosis, it's quite small at maturity, only 12 to 14 centimeters, smaller than the largest larvae. And again, because it doesn't feed after metamorphosis, it is the most cryptic of the three species, spending almost all of its life cycle buried in the sediment, and only really comes out shortly before spawning. Northern brook lamprey has the most restricted distribution in Manitoba, found only in the Birch and Whitemouth rivers. Because it's so cryptic, we thought that it may have had a wider distribution and had gone undetected, although our environmental DNA monitoring hasn't found it elsewhere. So it does seem to have a very limited distribution. So I mentioned that all three species are currently data deficient by CSIWIC, but the status is currently being reassessed. So we're hoping that they'll eventually be rescued from that data deficient category. Okay, I just want to wrap up before I take questions by first acknowledging the funding sources that have enabled my research: Faculty of Science, NSERC, Fisheries and Oceans Canada, Genome Canada, Great Lakes Fishery Commission, and they aren't mentioned by name here, but the dozens and dozens of students and colleagues who I've worked with over the years. Before I start questions, again, I'm just going to indicate here I've got my email address in case you want to follow up with any questions, or if you have some information about lampreys in Manitoba, or if you would like a copy of some of the book chapters upon which this talk was based, please don't hesitate to contact me. Thank you.
Great. Thank you so much, Margaret, for a very interesting talk and research. So just to remind everyone to ask your questions, please go to slido.com and event code 2521. Let me try to bring up the questions. Just a second. Hopefully that will show up. Okay, so here's a question for you, Margaret. I don't know if you can read it, but yes. I'll also read it. Do individual species, parasitic lamprey, have primary host species they prefer, or do they latch onto whichever species they encounter first?
Yeah, that's a good question, because I said that they weren't particularly choosy. That the sea lamprey, for example, has been reported on at least 50 different host species. Having said that, there do seem to be some preferences. So in the Great Lakes, lake trout seems to be the preferred prey. So some, at least during the larger parasitic stages, when they first come out of the rivers, they feed on smaller hosts and then sort of work their way up. So there seems to be host selection based on body size of the host, habitat. The ones that they come into contact more frequently will be their preferred in that sense. Silver lamprey, for example, really tend to like paddlefish and sturgeon, where you don't have, where you have a lot of naked skin to make it easier for attachment. So there certainly are some preferences, even though they aren't particularly choosy. And parasitic lampreys also make good use of invasive fish species. So again, they do seem to take advantage of what's there, even though there are some preferences.
Great, thanks. I forgot to mention that was a question from Josh. Thank you. The next question is from Hugo. Are other lamprey species present in the Great Lakes besides sea lamprey? If so, how are they affected by the measures put in place to deter sea lamprey?
Yes, there are. There are 4 native lamprey species present in the Great Lakes, so that the 3 that we have in Manitoba, plus the American brook lamprey. And unfortunately, there are negative side effects of the sea lamprey control program on the native lampreys. The lampricide that's used is largely lamprey specific. That it very fortunately doesn't have very many negative impacts on other fishes, because the other fishes are able to detoxify it. But it is lamprey specific, and so there is a certain amount of selectivity that's possible by where the lampricide is applied, so that the 2 brook lamprey species, the northern brook lamprey and the American brook lamprey, they're the non-parasitic ones, the non-migratory ones. So they tend to be found above barrier dams, and so that because those barrier dams prevent sea lamprey from accessing those upstream reaches, those areas aren't treated by lampricide, so that the brook lampreys then escape lampricide treatment. The other effects, the effects of barrier dams and lampricides in the downstream reaches, do affect the silver lamprey as well, and the chestnut lamprey. So some of the work that we're doing is trying to help sort of, and what the Great Lakes Fishery Commission itself is doing is trying to further increase specificity of the lamprey control program.
Okay, great. Next question: What grew your interest in lampreys?
Yeah, it all started with that one of my instructors, one of my professors at the University of Guelph, Dr. Bill Beamish, worked on lampreys, and they kind of sounded interesting, and I started working on them. Mm-hmm. I think the first 6 months it was sort of with a goal to eradicating them in the Great Lakes, and then the more I learned about them, the more I sort of grew at least a grudging respect for them, and then it became more than that. They just are so different than any other vertebrate, and at least when I started working on them 35 years ago, they were kind of the underdog, that there weren't a lot of people working on lampreys, and now, as I hope you've gotten a sense from my talk, that they've just become such an important scientific organism on so many levels, that I'm certainly very pleased with my choice.
Great, thank you. Next question. Besides the control of lamprey populations, what else do you research?
Yeah, a lot of my research is lampreys, both from a control point of view and from a conservation point of view. But other aspects of my research, especially now, involves using environmental DNA for other species of interest. And so I'm part of a large-scale Genome Canada project that is using environmental DNA for monitoring basically all freshwater fishes of Canada, again, sort of from a conservation point of view. So we've been using eDNA for monitoring carmine shiner and developing eDNA for lake sturgeon, but then also for monitoring invasive species using eDNA for zebra mussel detection as well. So those are sort of the main things that I'm doing now.
All right. Next question. This keeps moving up because of all the voting on the questions, so I have to keep track going back up and down. So the next question from Darren, what's the evolutionary relationship between parasitic and non-parasitic forms?
That's a great question, and I've spent several years of my life exploring that, and I wasn't able to talk about it at all in the 20 minutes, but it's a really good question. The parasitic and non-parasitic forms are quite closely related, that you might think that all of the parasitic ones are closely related to each other and the non-parasitic are sort of on a separate branch of the family tree, but that's not the case at all. In fact, in most lamprey genera, you have these pairs of parasitic and non-parasitic forms that are virtually indistinguishable as larvae. They're, as far as we can tell, genetically indistinguishable, and then at metamorphosis, one goes one way and feeds parasitically, and the other just skips the parasitic feeding phase and becomes sexually mature. And so the northern brook lamprey and the silver lamprey that we have in Manitoba are paired species, where indistinguishable as larvae, and then one feeds on lake sturgeon and pike, and the other doesn't. And in fact, there's quite a bit of debate about whether they're real species, whether they're actually distinct species, or whether they're just different ecotypes of a single species. Something I hope to resolve before I retire, but it's still very much an unanswered question.
Great, thank you. Next question from Janet. Thank you for the interesting talk. Can lampreys attach to humans?
There are reports of them attaching to humans. They generally don't. I think for the most part, lampreys, they must, I think that they can detect the difference between cold-blooded and warm-blooded hosts, but I have heard accounts of long-distance swimmers in the Great Lakes, once presumably their external body temperature has gotten quite cold, where they have actually had lampreys attaching. So there are some stories, for example, in Corey Brandt's book, about some long-distance swimmers in the Great Lakes that have had to sort of fend off sea lamprey while they've been swimming.
Great, thanks. Next question from John. What do you think might be some of the most exciting future applications to which research in your field might lead?
I don't know. Yeah, some of the stuff that one of the things I think sort of relates back to, I think, the first or the second question about whether or not sea lamprey control methods negatively affect the native species. I think one really exciting future sort of application is being able to further to make the lampricides even more specific, and so that right now that they're lamprey specific but not sea lamprey specific. And so there's a project that I'm involved with, led by Ken Jeffries in our department, using transcriptomics, looking at gene expression in sea lamprey to see if we can learn more about the mechanisms of toxicity of the lampricide, so to see whether it can be sort of tweaked to make it even more sea lamprey specific. So that's one area of research. And then another, which I'll try not to go into in great detail, but I'll at least mention, is sort of harking back to my PhD work, looking at sex determination in lampreys. When I started my PhD in the 80s, I thought that in my 4 to 6 years, I'd be able to solve all the questions related to lamprey sex determination. I wasn't, and it's still unknown. One of the things that has been observed in lampreys is the sex ratio, at least in the Great Lakes, during the initial invasion, seemed to be related to population density. When density was really high, the population was male biased, and then when the numbers came down after sea lamprey control, it shifted to a female biased sex ratio. And so I've been looking at environmental sex determination, whether population density influences sex determination. And so now one of the projects that I'm working on with postdoc Phil Grayson, a colleague Colin Garraway, trying to use genomics to find the genetic basis of sex determination. In some pest species, manipulating sex ratio, for example, making them all males or making them all females, is a way to control pest species. So those are some of the big picture stuff that I'm working on.
Excellent. Next question from Brianna Caldormer. Does lampricide used to kill sea lamprey kill native ones as well? Has there been an effort to develop one that only kills sea lamprey?
Yeah, so the lampricide unfortunately does. It's lamprey specific, but not sea lamprey specific. And the reason why it's doesn't kill other fishes is other fishes, the more highly evolved ones, if I can say that, are able to detoxify the lampricide, and lampreys can't. The effort to develop one that only kills sea lamprey, it's certainly, you know, desirable. The fact that this lampricide was found at all that was specific to lampreys is almost too good to be true. And again, sort of harking back to Corey Brandt's book, going through this, there were, like, I think 6,000 chemicals that were screened, and it was just by good luck in a lot of ways that there was one that was found that killed only lampreys and not other fishes. And so since then, we haven't been able to improve on that, but there certainly is work being done to see whether they can be improved to make it species specific and not just lamprey specific.
Great, thanks. The next question, I guess it's related to something you've answered before. Question from John. How did you decide that lampreys were your thing? That they were the ones to which you would dedicate your life's research?
Yeah, it was one of those things. I just sort of fell into it. One of my profs at Guelph was working on lampreys, and then it just suited me. And it's and one of the things I'll add that is different than what I've said already is it's one of the things I also really enjoy is it's a really good research community that everybody who works on lampreys is really interested in lampreys, whether they're trying to kill them or whether they're trying to save them, depending on the species in the ecosystem. But it's certainly a really sort of dedicated, vibrant group.
Great. And I guess Manitoba is a good place for that research, right?
Yes, yes.
That's great. Next question from Victoria Winters. What's your favorite instance of lampreys in pop culture?
I think I'd probably have to say, and I sort of gave a little shout out to it in one of my slides, the movie Blood Lake. That if you like good bad movies, it's a fun movie. That it's a story of sea lamprey attacking a town. It's a fun watch.
Okay, great. The last question we have for you, Margaret, from Ashok Dhawan. Do the species found outside Canada, I guess, are they found outside Canada, places where they are found the most?
Yep, pretty much. So that lampreys are found worldwide in temperate, the cooler regions of the northern and southern hemisphere, so that they're absent from the tropics because they don't do particularly well in warm water, although there are 2 species found at high altitudes in Mexico. But apart from that, if there is freshwater streams for spawning lampreys, and for the larval lampreys to live, you will find pretty much lampreys, so that they're throughout North America, absent the tropics, Europe, Japan, Korea, China, Iran, Russia, in the southern hemisphere, New Zealand, Australia, South America as well. That's pretty much anywhere you have fresh water, and then minus that strip in the tropics.
Okay, excellent. That ends the question period, Margaret. Thank you, everyone, for all your interesting, excellent, very engaging questions. Thank you, Margaret, for your wonderful talk. So I'll clap on behalf of everyone watching you online. And so that ends your part of the session. So I'm going to try now to bring up the next talk ad. If you can bear with me. Hopefully that works. Great. So yeah, just before we end today's session, I would like to advertise our next talk, which would be actually the last talk, spot talk for the summer series, and it's by one of our alumni, Nirvani Umadat, who's a dental surgeon, biotechnologist, and healthcare entrepreneur. And she's going to talk about the human holobiome, the biological communities that make us who we are. So I just hope to see many of you next week. We look forward to that. And just to remind everyone that all the talks are posted on the Faculty of Science YouTube channel. So if you missed part of a talk or if you want to go back and watch previously missed talks, they're all posted on YouTube, and you could follow them from the Faculty of Science Spot website, which is shown here at the bottom of the slide. So thank you everyone again for making us part of your day on a Friday afternoon. And I wish you a wonderful weekend. We'll see you next Friday. Bye for now.